Hello student, we are here for our remaining part of the bacterial plant disease. So this is the bacterial plant disease part second. Okay, in this point or in this part we have to study the black arm of cotton or vein blight. Okay, the causal organism of this black arm of cotton is Xanthomonas malvacearum or Pseudomonas malvacearum. This Xanthomonas bacteria contains the order Pseudomonadales and family Pseudomonadaceae. Then, as we know, this pathogen affects the cotton plant or cotton crop, and the cotton is very commercially very important in our India or cultivated in large scale in India. The annual production of fiber is about 5 million tons, okay, and 200 million tons per year. Hence the black arm of cotton is also called as the angular leaf spot or vein blight. Okay, it is less severe. Then the causal organism Xanthomonas malvacearum, it is also named as the Pseudomonas malvacearum or Phytomonas malvacearum. This bacteria is rod shaped, then motile, and it formed single or in pair with single flagellum. It is gram negative bacteria. It is non-spore forming and it is measured about 1 to 1.2 into 7 to 9 micron. It is without capsule and produce a yellow slimy colony on nutrient media. This is about the causal organism. This causal organism is known by these three names: Xanthomonas malvacearum, Pseudomonas malvacearum, and the Phytomonas malvacearum. As these all bacteria are generally rod-shaped structure, motile, and this bacteria is motile and it forms single or in pair with single flagella, and it is gram negative bacteria. They form their colony by forming yellow slimy in nature on the nutrient medium. Then the next point is the symptoms and signs. The first symptom is the bacteria attack on aerial part of the cotton plant. The first symptom is found on seedling. Okay, the seedling of this cotton plant first affected. The cotyledon in the form of water soak circular lesions, which later it become irregular and enlarge. They become brownish in color. It produces distortion and falling on cotyledons. Further seedlings also fall down. Newly formed leaves are also affected. On the matured leaves, water soaked lesions first appear. They become angular bound by veinlets and turn dark brown. Size is about 1 to 5 millimeter. As the first symptoms show on the seedling, this pathogen first attack on the seedling. After affecting this cotyledon is looking like the water soaked circular lesion, or later it become irregular or enlarged. Okay, then they become brownish color. The cotyledon is looking like we are soaking this cotyledon into the water, and then it turns enlarged and dark brown in color. Then newly formed leaves are also affected. Mature leaf where soaked lesions first appear on mature leaves, and then they become angular bound by veinlets and turn dark brown to color. Then the size is about 1 to 5 mm. The many spots unite to produce irregular black patches. To spread this disease, these many spots are getting connected or they getting united and form the large black patch with become dry. Mainly this disease is spread by the veins, and hence it is also called as the vein blight. As this bacteria cause water soaked lesion, hence these lesions at first they are light brown or dark brown, and then these lesions turn into dark black in color. Then On the stem and fruit, it forms dark brown to black colors of sunken lesions. Then these bacteria girdle the stem and the branches and make them drop off. These spots are deeply seated and it spreads inside ball and infect the fibers. In some area, loss it may be up to 80%. As I say, this disease is spread on the cotton balls also, then cotton fiber and cotton fiber also. Hence, in some areas, it it made up to 80% of loss. And the cotton is our commercial cultivation or commercial crop variety. Hence, this disease is important as a pathologically. As we know, this disease is seed-borne and primary infection start from the seed, where the bacterium may present as a slimy mass in a seed. Hence, we have to first treat the seed. Okay. First control measure is to removal and destruction of disease host as well as plant debris and remove and burn. Then crop rotation, then late sowing, early irrigation and addition of phosphate fertilizer helps to reduce the infection of this disease. Then the primary infection from this seed-borne bacteria can be checked by treating seed. Okay, we have to soak the seed with concentrated hydrochloric acid or 1 to 15 millimicron organomercuric chemicals. Then internal infection can be eradicated by soaking the seeds overnight in 1000 part per million of streptomycin sulfate. This treatment of seed is very important in this case because this disease is spread from the seedlings first. So we have to treat these seeds. Then spraying of fungicides like diethane reduce the disease intensity. Use resistance variety which are limited in success: Gossypium hirsutum, Gossypium barbadense, Gossypium herbaceum, Gossypium arboreum. These are some resistance variety of the cotton crop. Studies for the resistance variety are still in continue. We can also control this disease by sowing the seeds or storing the seeds for 1 year and sowing these seeds after 1 year. This method is found successful or useful in the control of the citrus black arm of cotton.